Hello everyone and welcome to today's webinar brought to you by LGC Axio Proficiency Testing. It's my great pleasure to welcome all of you who are joining us today for our session entitled Environmental Evolution, the Past, Present and Future of Analytical Science in Neonicotinoids and PFAS Pollutants. The subject of pollutants is an ever-prevalent topic of conversation, one of which continues to evolve, and one we are excited to explore with you today. So thank you for joining, and we hope that you find today's webinar informative and valuable to your work and your knowledge base. So just a short introduction from myself. I'm your host for today, and my name is Brooke Dawson, and I'm the Global Assistant Brand Manager here at LGC Axio Proficiency Testing. But more importantly though, I'm delighted to be joined by our very own Axio expert that we'll introduce in just one moment. So, what is this webinar about? In this session, we will explore the importance of proficiency testing in environmental science, whilst focusing on two emerging pollutants of environmental concern, neonicotinoids and PFAS. We will look at how history has impacted today's regulatory legislation and how those requirements have advanced as analytical testing methods for these substances has evolved. We will consider the trends in the Axio AQUACHECK scheme, providing real-life examples of proficiency testing data within the scheme. And finally, we will look at the future of analytical testing in pollutants and what that could mean for you in your laboratory. So today I am delighted to be joined by Dr. Matthew Leeton. Matthew is the Head of Technical Operations for the Proficiency Testing Group at LGC. He is responsible for the production, development and technical operation of our PT schemes in a diverse range of analytical fields, including the water industry, soil and contaminated land, food, animal feeds, pharmaceuticals and consumer safety. Prior to joining LGC in 2007, Matthew spent more than 10 years in analytical chemistry, specialising in the analysis of pesticides in food and environmental matrices, so he's well equipped to talk us through this hopefully insightful session. So before I hand over to Matthew, just a quick note, there should be some opportunity at the end for some questions, so if you do have a question as we run through this session, please do write it in the chat function in GoToWebinar, which is at your right-hand side. And we will try to tackle as many as we can at the end. So I think that's enough from me for the time being. So I'm going to hand over to Matthew to start our presentation entitled Environmental Evolution, Past, Present and Future of Analytical Science in Neonicotinoids and PFAS Pollutants. Matthew?
Thank you, Brooke. Welcome to everyone taking part in the webinar, and it's my pleasure to take you through the information that we have regarding specifically neonicotinoids and PFAS, but touching on some general aspects of the advancement of testing and where proficiency testing can help participant laboratories to improve their data. Neonicotinoids are a group of insecticides which are used on an extremely wide range of crop types and can also be used to control a variety of pests, but particularly and primarily sap feeding insects such as aphids and root feeding grubs. They also have veterinary uses for tick control and flea control in pets. The name neonicotinoids, which has been shortened in various places to neonics or NNIs, literally means new nicotine-like insecticides because they have chemical similarities to the molecule nicotine. They have been registered now in more than 120 countries around the world, and in 2008 the turnover of the sales of these chemicals was more than 1.5 billion euros, which is approximately at that time 24% of the global market for insecticides. So in those times, they are certainly a major player in the insecticide market. In terms of the neonicotinoids themselves, you can see the structures of several common molecules there. And they can be classified in 3 different ways. The N-nitroguanidines, which includes perhaps the most well-known molecules, imidacloprid, thiamethoxam, and clothanidin. And then N-cyanoamidines, which covers acetamiprid and thiacloprid. The neonicotinoids are very useful as pesticides and insecticides because they're systemic pesticides, which means that rather than like the classical contact pesticides, they don't remain on the surface of the plant, but they're taken up by the plant and they're translocated through all of the organism throughout all of the plant to leaves and roots and various parts in order to have their effect. And that makes them very effective as pesticides, but also gives certain complications when we're considering how the neonicotinoids can function as environmental pollutants. In terms of their mode of action, they work by affecting nicotine acetylcholine receptors, which are part of the insect central nervous system. It causes excitation within the nervous system of the insect, over time causing trembling, followed by paralysis, and followed by death of the insect itself. This affinity for the insect nervous system actually means that they're very good at non-targeting for mammals and human beings. So they have a very poor penetration of mammals and certainly the blood-brain barrier, and so neonicotinoids are very non-toxic to mammals. One of the prime reasons that neonicotinoids are systemic fungicides or systemic insecticides is their water solubility. This helps them to move around the plant because plants use water for their systems to move materials within their system. So neonicotinoids are generally water soluble. The actual solubility of the neonicotinoid compounds depends on the individual compounds and depends on their structure. But it also means that the application of the pesticides is particularly important. It has a huge effect on the rate at which these pesticides can move into the water system and the aquatic system around the areas of application. And it's been shown that the primary mechanism for transport of neonicotinoids into water bodies actually occurs very shortly after or during the spraying season via runoff. So a lot of the environmental pollution caused by nicotinoids is caused during that planting season. Also, the neonicotinoids can be relatively stable. If they're susceptible to photo degradation and under natural light, the neonicotinoids will break down fairly rapidly, and particularly as well under alkaline conditions. But because the nature of the materials means that they're not always exposed to photo degradation very frequently or for a very long period of time, they may persist in soils for a very long period of time. So as we've seen, there are a number of concerns that we have or that have been raised about neonicotinoid insecticides, and that is clearly the idea that they're systemic pesticides. The physical characteristics that neonicotinoids have to have in order to be systemic give them a lot of properties which make them also very adaptable as environmental pollutants. They're rapidly translocated through the plant. They get to all parts of the plant, and so they can affect organisms which either eat or live within all of the plant tissues. They're very rapidly translocated. They get a very high take-up rate. So once they've been applied to the plant, a large amount of the active ingredient is taken up by the plant, which is good when we're thinking about the amount of ingredient we actually need to use. But it does mean that it's taken up by the plant and it's not degraded as a contact pesticide would be. They can be very persistent. We've seen in various areas, particularly where we don't see photooxidation, so the chemicals are protected from sunlight. The clofanidin and imidacloprid residues can remain in the soil for a very long time, and certainly in the EU they've been classified in the very persistent category. And the same chemicals have been shown in the US to be active in the soil for longer than chlorpyrifos, which is a banned organophosphate pesticide, which has been a chemical of concern in the past. Clearly, because of their systemic nature, their water solubility, their ability to move in the aquatic environment, they have a high potential for leaching into water. This means that we can establish basically a reservoir of these pesticides in plants, in soils, and then as they're affected by water, or as water comes into the system, these pesticides can then leach out of the reservoir that's been created into the water. It's been well studied in the US, and it's been shown that neonicotinoid insecticides have moved from the areas in which they've been applied, and they've been detected all around in fields, in puddles, ponds, wetlands, rivers, snowmelt, and groundwater. So in certain areas they're almost ubiquitous in the environment, and because of this ability to travel, they can be devastating for environments, and their toxicity to insects, both beneficial and target insects, means that they're very effective in terms of removing them from the environment. And this idea of non-target contamination is perhaps the most well-known and well-understood thing about neonicotinoids, because the effect that neonicotinoids appeared to have and then were studied to have in bees was the primary reason that neonicotinoids have been most recently legislated against. We can see from the history that neonicotinoids have had a stunning rise, and then most recently a bit of a dramatic fall. They were, as we've said previously, they were developed in the 1980s. The first available compound, imidacloprid, has been used and available to farmers since the early 90s, and approved by the EU in 2005. So for agrochemical, that's a fairly quick movement throughout the the world in terms of getting approval. And then as time went on, once imidacloprid was approved for use as a plant protection product, then other chemicals which were related to imidacloprid were also approved. So clothianidin, imidacloprid, thiamethoxyamacetamiprid, and thiacloprid were the chemicals which eventually got approval by the EU. As I said, one of the key things that was observed as these compounds became more and more popular, more and more widely used, was a clear effect on bee populations. Well, non-target organisms, but specifically honeybees. And it wasn't long, a mere sort of 8 years after the introduction of these compounds in the EU, that restrictions were placed on the neonicotinoids to stop their use and to restrict them to use under specific conditions and in specific ways, to try and protect honeybees and prevent them from suffering further losses. This has resulted in 3 of the active substances being banned in 2017 for outdoor uses, and then further restrictions in the EU have been were placed in 2020 in order to basically eliminate the use of these compounds throughout the European Union. At the same time, though, their use as highly effective insecticides was recognised for emergency uses on specific crops. Probably most notably is sugar beet, which is a very important crop across the EU, and so some emergency authorisations still remain in place in Europe, probably until another plant protection product is identified which is suitable for the role that the neonicotinoids are being used for. Across the United States, the decisions haven't been quite as dramatic. The 5 neonicotinoids that we've been talking about remain in use and remain authorized for use in the US, although there are restrictions in how they may be used in order to try and prevent overspray, to try and prevent runoff and leaching into watercourses, and generally protect honeybees and other pollinators. These decisions are due for review fairly shortly, and it may be that that further restrictions on the authorizations for these compounds are applied. So that's a bit about the background of neonicotinoid insecticides. The other group of compounds that we want to really touch upon today is the so-called PFAS, or polyfluorinated alkyl substances. I think these are very different to the neonicotinoids in that when we think about polyfluorinated alkyl substances, I'm definitely going to call them PFAS from now on. We're not thinking about a very specific group of compounds and a very specific number of compounds. PFAS are synthetic compounds. There's around 4,500 of the different chemicals which would fall into that PFAS category, and they have arisen over time to be some of the most persistent and some of the most widely found compounds in the world to date. Probably the 2 most commonly known are perfluorooctanoic acid, PFOA, and perfluorooctane sulfonic acid, PFOS, mostly because these compounds are used in a wide variety of consumer products. So I think all of us have at some point owned or do own a pan with a non-stick metal coating to ensure that the food doesn't stick, and typically those coatings will contain PFAS or similar molecules to give them that non-stick property. But it's not just cooking pans. The range of products in which PFAS are intentionally used are huge, including food packaging, firefighting foams, cosmetics, and as we see later, that applicability to many different areas of life is one of the reasons why they've become such an environmental problem. So this is I guess you could call it a general structure of a PFAS molecule. In fact, this is PFOA, but the most widely accepted terminology for PFAS and definition of PFAS is that they are synthetic chemicals. These kind of things don't occur in nature, so I think anything with this kind of structure, it's a given that they're synthetic. They contain one or more carbon atoms, and all of the substituent groups on those carbon atoms, which would typically be hydrogen, have been replaced with fluorine. So this is what they look like. The bond between carbon and fluorine is perhaps one of the strongest which is known, so it makes them very, very resistant. It means that they don't degrade easily. There's not many things that can attack them in nature, and therefore if they can't degrade, they can easily get into a position to accumulate in the environment. And this is why PFAS have been over the years labeled as forever chemicals. The environmental concerns with PFAS are widespread. There are a number of ways in which they can have effects on the human body. As we've said, because of their persistence, because of their ability to accumulate in the body, the PFAS molecules are found pretty much everywhere. We find differences between the PFAS molecules themselves. So the smaller chain PFAS molecules are much more likely to be water soluble and distributed in aquatic environments, whereas the longer chain substances, because they tend more towards fat solubility, they have the potential to accumulate in the organs of things like seabirds or aquatic animals. The problem with one of the problems that we've seen with PFAS is the huge number of compounds that fall into that category. If we're thinking of 4,500 compounds, it's very difficult to carry out specific risk assessments and to manage risks for all of those chemicals individually. And because we can't control them individually, we now need to think of them as a class of compounds and carry out our control on that class of compounds as a whole. The PFAS chemicals, as we've seen, because they're stable, because they have the potential to accumulate, that would be a bad enough thing on its own. But also, they are or have degradation pathways, which leads them to be chemicals which can affect human beings, can affect the unborn child, and can be found in affecting human beings in numerous ways, many of which are well understood, but some of which aren't. And it's been shown in 2015 that over 95% of the adults in the USA now have some amount of PFAS in their blood serum, and that is very unlikely to change over time because of the rate of breakdown is so slow. In terms of the history that we have with PFAS, they were discovered by accident in the 1930s. DuPont were looking at new chemicals which could be used as refrigerants, and then they produced a new substance by accident, which was coating one of their test chambers, and that substance was PTFE. Again, a very hugely and widely used chemical now, and so this has been now marketed and used across a range of products. A few years after that, 3M discovered Scotchgard, which was a PFAS chemical, all of their very own. In this case, that was PFOS. They were trying to produce a rubber which wouldn't deteriorate, but instead produced a chemical which was highly resistant to water and alcohol, and then wouldn't be removed very easily from the material that it was attached to. So Scotchgard came into being. The uses broadened out as time went on, and in the 60s and 70s, and the US Navy base fire expanded their use as aqueous film forming foam, which is a mixture that rapidly extinguishes fire because it's not easily degraded, and it can expand into all the places to eliminate oxygen from the fire. So we're now seeing this uptake of PFAS into numerous areas of our lives, and which has made them the ubiquitous chemical that they are today. I think the first signs that PFAS may not be the wonder chemical that we were hoping for over time started to come with scandals around the DuPont factory in 1999, where PFAS, which were being dumped into water courses around the Ohio River near the DuPont plant, were contaminating drinking water supplies of around 100,000 people, were responsible for killing cattle, and it's been shown that the PFAS exposure from these chemicals have been linked and associated with kidney and testicular cancers, reduced immunity, etc., etc. So the effects of these chemicals which have escaped into the environment is very significant. The PFAS chemicals have now been listed under COP4 in 2009, where the conference parties listed PFOS, PFOSF, PFOA, and its salts. The PFOA has been banned in the EU under the persistent organic pollutants regulation. So we're starting to see more and more countries restricting the use of PFAS compounds and banning them outright in some cases. Within the environment, the Drinking Water Directive in the EU has set standards and has set maximum allowed concentrations for PFAS in the environment. The limit value for for PFAS is 0.65 nanograms per litre in surface waters, and then 0.13 nanograms per litre in seawater. So the idea is to make these materials very restricted and only present at very low concentrations. So we've got into a bit about why, what the chemicals are, and why they present a problem, and why they present an environmental concern for human beings. So let's have a little bit of a look at the monitoring strategy and the measurements which can be applied to these groups of chemicals. In terms of neonicotinoids, they've been included on the EU watch list in 2015, which meant that all countries within the EU had to look for 17 specific compounds, of which neonicotinoids were 5, and they would monitor a minimum number of sites at least annually for these chemicals. So this was done, and the results came out a year later. And if we looked specifically at the UK's data for this, 23 sites were sampled in 2016 for imidacloprid, clethianidin, thiamethoxamacetamiprid, and thiacloprid. 88% of the samples which were analysed contained compounds of interest. That covered 17 of the 23 sites. The neonicotinoid concentrations ranged from 2 nanograms to 443 nanograms per litre, and 8 of the rivers that were monitored in the UK exceeded the recommended chronic pollution limits. So it was amazing how widespread these chemicals were within the UK, when actually the use of those compounds seemed to be quite unlikely. So imidacloprid was detected in a number of arable river catchments, despite it having very little use on arable fields in 2016 or the preceding few years. So that speaks again to this persistence and this bank of contaminant that has been built up over the years. In other countries, numerous authors have reported studies of neonicotinoid monitoring. It's been a very widely reported subject. A review of 29 studies from 9 countries found that neonicotinoids, as with the UK study, were detected in the majority of water samples. Typical concentrations, again, were around 0.13 micrograms per litre or 130 nanograms per litre. And that was the average concentration. It was found that peak concentrations, which were taken shortly after the planting or shortly after application, could be up to 0.63 micrograms or 630 nanograms per litre. In the US, the environmental campaigners in the US want the EPA to follow the European Union banning 4 of the neonicotinoids which are considered to be most damaging. But as we said previously, it's likely that the EPA is going to hedge for more restricted use rather than outright bans in those cases, which could potentially give neonicotinoids approval for another 15 years. In terms of PFAS, again, a huge amount of work has been undertaken to look at the analysis of PFAS in various countries and in various studies. One of the key problems with PFAS is that there's a huge number of compounds, so it's a very complex area in terms of testing, and it can be quite difficult to ensure that comparable methodologies are being used in all of the studies. In terms of what we want to get out of it, we need to have accurate and robust analytical methods for the quantification of PFAS, and that's part of the rationale for developing proficiency testing to support the method development which is currently ongoing for PFAS. A number of different techniques have been applied over the years for looking at PFAS. Methods which look at individual compounds, so things like LCMS, methods which are going to then sum those individual components, screening methods which are looking at fluorine, so total fluorine or extractable organofluorine. But we need to understand what people are measuring when these various methods are being carried out. The limits of detection that can be achieved are typically around the 0.5 microgram per litre range, and you can see that the regulatory limits for each of the materials has been listed. So the blue, sorry, the red dotted lines represent the limits from the Drinking Water Directive, and the blue lines are the values for the Water Framework Directive. So we can see that frequently the maximum value that's been reported exceeds those regulatory limits, particularly in surface and fresh waters, by a considerable amount, given the logarithmic scale of these graphs. So these compounds are being found, and they're being found at significant levels. So how is this testing being undertaken? It's clear that there's an analytical challenge. With neonicotinoids, we can see that one of the challenges is the relatively speaking, the low concentration of neonicotinoids in environmental samples. They're highly effective pesticides, so they don't necessarily need to be present at very high concentrations to create an effect. Therefore, the concentrations that we need to measure down to need to be relatively low. So we're typically looking around the 0.1 to 10 nanograms per litre for the values that we expect to find in environmental samples. When we're thinking about analysis at those concentrations and at those levels, typically we're applying some extraction methods, which are then going to give us the potential to remove interfering species, but also, and probably most importantly for a number of these assays, is to increase the concentration of the analytes that we're looking for in the sample. When we're thinking about these extraction methods, it's been reported that pretty much every analytical extraction method available has been applied to neonicotinoids at some point, with probably the most common being SPE, or solid phase extraction, but also things like liquid-liquid extraction, dispersive solid phase extraction, and then micro extraction techniques have also been applied. Once we've completed the extraction, then unsurprisingly, the method of analysis is pretty much whatever the lab has had available. So it's possible to use gas chromatography for the determination of neonicotinoids, but probably more commonly these days is the use of liquid chromatography. Both techniques can be used in order to detect all of the parent molecules and significant metabolites. A lot of the techniques that are used include mass spectrometry as the detection method of choice, purely because this gives better sensitivity for the compounds of interest. Though for gas chromatography, an electron capture detector can also be used. We've seen in cases where the matrix is particularly clean, where there's not a lot of matrix effects, that LC-MS can be used with direct injection, which is an excellent technique in terms of reducing the amount of sample preparation which is required, and therefore can give a better precision for the methodology. The detection limits that have been applied using these techniques, again, are typically in the 0.1 to 1 nanogram per litre range, although we'd expect some of the metabolites to maybe be a little bit higher than that. The methods have their positives and negatives, because with neonicotinoids, we're not just looking at environmental samples as aqueous samples. We also want to understand where the nicotinoids are in the plants, where they are in soil. We may be looking at a range of different techniques in order to cover all of the matrices of interest. And because we're looking at multiple matrices, it also means that they're amenable to different analytical techniques. So GC techniques, largely GC-MS, commonly applied for multi-residue screening, of which neonicotinoids could well be included, and they would be applicable for food or sort of plant-based matrices. So it may be that's applicable for some of the solid matter analysis. In terms of LC methods, because we're considering quite polar molecules in some cases, LC-MS is probably the method of choice for aqueous matrices. And as we've mentioned previously, clean water samples with modern equipment and new technology may be analysed without any sort of pre-extraction or cleanup, so a direct injection analysis. One of the things that we're seeing increasingly is non-targeted screening, and this, as you can imagine, is particularly interesting where we're looking at a disperse and diverse range of compounds like PFAS. Non-targeted screening is a very beneficial technique because we literally don't need to know what is in the sample, and we can find anything which is in there. The idea that we don't know what's in the sample is both beneficial and it can also be a downside. We can look for hundreds, if not thousands, of chemicals in one run using a technique like high resolution mass spectrometry or via TOF or QTOF mass spectroscopy, and we can know that a chemical or a material of a certain mass has been found. It may be that's a hit when we look at some software for deep pre-processing and some screening lists, and it may be that we can then identify what it is. But the downside of non-targeted screening is that probably 9/10 of the compounds that we're going to identify or that we're going to detect are not going to be identifiable. So we need to build on the screening lists that we have and the databases that we have to support our ability to measure so many compounds. And also, once we've undertaken non-targeted screening, if we want to then quantify things, in many cases we'll need to carry out additional analysis to support the quantification of some of the compounds that we've detected. It's clear, though, that the non-targeted screening is something that can be used for PFAS because we're not restricted to looking for just a handful of compounds out of the 4,500. and we know that there are not reference materials available for even 10% of those compounds that could enable us to do traditional make up traditional standards and carry out traditional analysis protocols. Many companies, many laboratories around the world are now setting up a screen of around 40 PFAS compounds, which is being proposed for monitoring purposes. They cover a range of PFAS molecules, which means that we can kind of extrapolate the total PFAS concentration from the measurement of those particular compounds. We can carry out a total PFAS measurement, so using something like absorbable organic fluorine. So this is a combustion technique which gives us a measure of the total concentration of carbon fluorine bonds within a sample. So it's not compound specific, but it does give us a good indication of the total PFAS loading within a sample. If we do want to go truly quantitative, though, we need to the gold standard is to spike with isotopically labeled standards. Again, we need to know that those materials are available, and they need to be produced to make sure they are available. And typically those analyses are going to be carried out by LCM SMS with confirmation by multiple daughter ions and recovery correction to the isotopic standards that we've measured against. So we've talked about how the PFAS and neonicotinoids might be measured, but then we need to look at how those methods are performing, ideally how they're comparing from one to the other, and how close they are getting to the true answer and the correct answer. And probably the best way that we can do this is by proficiency testing. So within Axio, we do run a number of PT samples for pesticides. So we cover the analysis of common insecticides, herbicides and fungicides. Of the methods that we're seeing within environmental samples, particularly aqueous environmental samples, over the last, say, 10 years, we've seen direct aqueous injection, LCMS, become the predominant method for this type of analysis. Again, we're talking about compounds which are water soluble and samples which have quite a clean matrix, and there's not a lot of interference in there. But also we've seen a significant improvement in the spread of data with the predominance of direct aqueous injection. We've seen a really significant increase in performance as a result of the application of a method with so little preparation and cleanup. Removing the analytical stages for extraction has had a very profound positive effect on the participants. This is just some more data. So again, showing the range of performance, the performance of methods, different methods within the sample is one of the key things that proficiency testing can offer. It's not a means of necessarily determining which method is the best, but it certainly has a good mechanism with enough data for comparing how each of the relevant methods will perform in a specific sample type. So just to touch further on the role of proficiency testing in environmental tests. Yes, the role of PT, proficiency testing in environmental testing. Well, proficiency testing it's an evaluation of participant performance set against pre-established criteria, and that takes part by an inter-laboratory comparison. What we're doing within proficiency testing is providing an infrastructure, providing a system by which the labs can monitor and subsequently improve the quality of their measurements. So we're not telling laboratories what to do, we're putting something in place to give laboratories the information on how they're performing, how their methods are working, how they're performing in comparison with other laboratories, with other methods, with other techniques, and then giving them a base on which they can build their quality improvements. The strength of proficiency testing is that it's independent, but also that it can be applied regularly. So labs can get an ongoing performance assessment. It's not just a one-off that's carried out when the method is validated. It's ongoing for the duration at which that method is run within the laboratory. PT is profoundly important if we're considering laboratory accreditation. It is a key in ISO EC 17025 in order to fulfill the requirements for getting accreditation. Having, again, it's that independent carrying out analysis on something that you don't know the answer to and performing successfully is a great means of determining that the method works as expected. PT can also be used to demonstrate laboratory competency to customers and regulatory bodies. More and more customers and regulatory bodies are knowing to ask the question, do you perform in PT? How do you perform in PT? We've talked about the comparative benefits of proficiency testing, but the key thing I think is to use PT to educate and to train. You know, both the staff and also the systems which are applied. So taking learnings from each participation within PT and seeing what we're doing well and seeing what we're doing less well is a very powerful tool. It can be used to, again, with this comparative exercise in mind, it can be used to verify methods and instrumentation to make sure that they're performing as equivalently as we think they are. And one of the things that it does do is it can flag up potential problems. If we look at trending through a regular performance in PT, we can look at things where things are starting to go wrong, which gives us a great way of being able to manage risk. Just a little bit about the AQUACHECK scheme that we've seen the data from. It is a very long-running scheme. It's one of the largest schemes that LGC Axio proficiency testing operates, and it covers areas of water, soil, sludges, and it's the leading water chemistry PT scheme that has been in continuous operation since 1985. In terms of the recent scheme year, 2021, the scheme had over 600 active participants across all of the groups and the test materials which are offered. So those cover things like major inorganic and organic contaminants, trace elements, organic compounds such as pesticides, phenols, herbicides, and many others. It's very broad reaching with over 40 different samples across the range of products that are offered. I think as you've all joined this webinar, you all regard water as being a very important subject, and environmental pollution and measurement of environmental pollution equally important. You know, water is clearly vital in making sure that natural systems are as supportive as possible. And each of the water sources that we're considering needs regular analysis to determine the safety and the suitability of use. So from potential leaching from drinking water pipes to presence of microorganisms and algal blooms in surface waters. We need to monitor these things to make sure that they're correct and safe for us to use. We need to make sure that we're not discharging harmful things into the environment through wastewater treatment plants, and also as a secondary thing that the industry is complying with environmental guidelines. We want to make sure that people are doing what they say they're doing and doing what they're allowed to do. We need to keep processed waters clean to ensure product quality, and we need to make sure that these waters are checked regularly to make sure that we're not missing anything, and that we're getting the quality of water in all the areas that we expect we are. And it's laboratories around the world who are making sure that this is the case. The quality of their analysis is the tool which supports our water supplies, whatever they are. And we, as AQUACHECK AXIO, we support those laboratories in ensuring that their measurements are correct. So participation in AXIO AQUACHECK scheme allows labs to identify the problems before the quality and the safety of waters are affected. Participants will be able to demonstrate independently that they're producing accurate and meaningful results. And if you want to discover more, you can visit lgcstandards.com/AQUACHECK or by scanning the QR code on the right now. So in addition to the AQUACHECK scheme, we have a range of other supporting schemes in the environmental area. As I said, we have our AQUACHECK scheme, which is covering water and agricultural soils and sludges. We have QWAS, which covers the same range of matrices, but for water microbiology. Our air PT scheme covers air and stack emissions. Our contest scheme covers contaminated land. Hygiene is a scheme which looks at surface monitoring for microbiology, so swabbing and plate count techniques predominantly. And then our crypt scheme looks at the analysis and measurement of cryptosporidium in water. So what's next? Well, in terms of the samples that are being provided within the AQUACHECK scheme, the next thing is our new neonicotinoids in groundwater sample, which is being produced. We're going to provide a groundwater sample and a spiking solution, which participants can or will combine in order to produce the test material. This is mainly because we want to ensure the stability of the compounds of interest by providing them at a concentrated level in solvent. And we'll include the 5 most common neonicotinoids as target analytes. So clothianidin, imidacloprid, thiamethoxam, acetamiprid, and thiacloprid. In terms of PFAS, we're intending to launch a PFAS PT sample later in the year. So if you want to stay in the know with that, you can click Get Updates, which will register you for our email newsletter, and that way you'll get the information when that sample launches. But also, if you have specific requirements that aren't currently met by our proficiency testing schemes, and that's certainly something that over the years a number of labs have done, you can suggest a new sample by clicking on the the link on the website and give us a description of your requirements and what kind of material you're looking for, and we will add that to our list of potential new products. So in terms of, just to give you a little bit of background on Axio proficiency testing, for over 40 years, LGC has been driving proficiency testing and leading its direction in order to make proficiency testing the technique that it is today, to help bring our technical expertise and influence and to push quality assurance in the direction that it needs to go. Our aim is to consistently design our schemes to meet your lab's current needs, but also the future needs. And so what helps you consistently deliver a quality product, whether that be actual products or in testing, is what we want to do in order to ensure that specific standards are achieved, or that accreditation to the relevant ISO standards is maintained. So we feel that great analytical results are born out of confidence, and by working with LGC Axio proficiency testing, you can have the assurance that your decision making is informed, is reliable, and is effective, because that's what your results are. So thank you very much for listening. We'd love to hear your feedback. If you want to discover more about LGC and Axio PT schemes, you can go to the website there, or you can contact us via email at axio.pt@lgcgroup.com, or join us online on LinkedIn and Twitter. Thank you very much for listening.
Yeah, thank you, Matthew. Hopefully everybody has found that session interesting. After this session has concluded, there is a survey available for which is 5 short questions. It's very quick to fill out. We'd love to get your feedback, and it also gives you an opportunity to make suggestions on future webinar topics. So what do you want to hear about? So we have had some questions through, although I'm very conscious of time. So what we'll do is those who have submitted questions, we will reply to you individually after this session, and we'll do that via email. So I just want to thank Matthew for his time today. Hopefully you've enjoyed the session, and thank you on behalf of LGC Axio Proficiency Testing to you guys for attending today. Thank you, and take care.